Professor Watson, we're of course very happy to have you here. I know that you already complained that you had to do too many things, but now for an hour or 1.5 hours we'll relax and go over your life. And we have a few pictures here. We also have a few video clips that we're going to see during the presentation. This is you. I don't know if you see it in here, but in any case, you know, right? This is you when you were young. So I guess we could start, since there are so many young people, young students, right? It says here that you only did 2 years of high school, right? And then you went immediately to the university. Is it correct? And first of all, what did you do when you were young? I know that you had an interest in ornithology, of all things. So you could tell us about that.
I think I was, when the weather was good, I was, watching spring birds. The most consistent thing, I think, of my life was reading newspapers. It was by the time I was 10, in 1938, it was the Spanish Civil War. And so my family was very strongly pro-English and believing we should join the English and fight Hitler. So there were sort of 2 great crises, the Hitler crisis and the economic depression. So my family, during the Depression, my father's salary went way down. We lost the use of a car. So we used public transportation. But I think was that I really was interested in the war. It was just dominating. Where were we going to win it? And the other, thing from my background was important was. Strong belief in science, a rejection of religion, and a belief that the science of biology would be explained in terms of science. So I wanted to be a scientist. My father had a brother who was. Became a physicist, so it wasn't completely unusual. But the chief thing I got from my family was just books and newspapers.
And I know that you don't like people who don't read newspapers or books, right?
I find boring. Yes.
Even if some of them is named Crick, right? I think that.
Well, to a certain extent. I mean, Francis did read, but he wasn't interested in English politics at all. But during the war, he was, though he couldn't talk about it, he was really he was a war scientist. He was an important one. He was in the anti-submarine program in the Admiralty, which at the beginning of the war attracted the brightest scientists in England went into this submarine effort. It was only when the submarine effort had won that they then went to America and joined the uranium effort. But the submarines had to be defeated, and Crick played an important role in that. So.
But we'll go back to Crick later on, actually, because before I wanted to ask you about this book, which is called What is Life? What was the effect or the impact that it had on you, and not only on you, also on Crick and a lot of other people, I think.
I think it was to make biology romantic. It was, it was more than drawing plants and animals, descriptive things. It was going to have to have at its heart molecules which carried information. And the question is, how could you copy the information? So that was the, and it was written by a very famous physicist, Erwin Schrödinger, who was in in England, who was in Ireland for a strange reason. The English wouldn't let him in because he was living simultaneously with his wife and another woman.
Yeah, too many wives.
And so they wouldn't give him a job in Oxford or Cambridge.
And not even in Princeton, because he was offered a job, I think, at the Institute, and he said, I would come with 2 wives. And they said, you know, they didn't like that, right?
So he did write this book. It was, it ended with a very strange, to me and most people, sort of mystical chapter about entropy.
Yeah,
and,
I became God, right?
Yes, so it became, you know, I didn't understand the last chapter, but it didn't bother me because chapter 4 posed the problem that I would then work on until we got the structure of DNA.
Did you ever meet Schrödinger?
No.
Uh-huh. But you did meet...
Francis Crick wrote Schrödinger a letter after we found the double helix and did get a reply from him. Yes.
But you did meet Delbruck, whose ideas, of course, were the main contribution to the book of Schrödinger.
Yes. And so I met him because he was a very active co-worker with Salvador Luria, and they had started working together in 1941. And so in taking Luria's course, my first term at Indiana, I quickly learned that Luria and Delbruck were already a famous pair.
Here is a picture, incidentally, for those of you who have never seen it, of Salvador Luria on the right. And this year actually is the first centennial anniversary of his birth. You probably know this, right? So he would have been 100, right? And tell us about the influence that Luria had on your life. For example, I remember I read once that you said, he was always ready to rescue me. In what sense? What did you do? Why did you need to be rescued?
Oh, I guess on certain occasions I appeared arrogant.
Oh, one guesses why.
No, I mean, there was a meeting for, the start of your thesis, what are the courses? And someone wanted me to take a course on histology. And he was on the committee. He taught the course. But I said I wouldn't learn anything that would be useful in my life. So
how would you know in advance?
Well, in histology, you don't look at things in terms of molecules.
Okay.
It's just, tissues. And, it was out of the era of biology where drawing and photography were the way you establish your reputation, not by thinking.
And so you came to, you actually went to Indiana, right, to study with Luria, right?
No, I went to Indiana because they had just appointed the world's really best geneticist, Herman Mower, to its faculty. Mower was left-wing, and in Texas, being left-wing in Texas was not a good idea.
Even now, I think.
And so he decided to go to Berlin, where there was a Russian Timofey Frozovsky, had moved from Moscow to Berlin. And but as soon as Mower got to 32, he got to Berlin. Hitler came to power, and so he moved to Moscow. And in 1936, he wrote a famous letter to Stalin in support of genetics, and it had just the opposite effect. Stalin started imprisoning the geneticists. So Mower effectively had to flee Russia, and had no sympathy at all for Russia, but was, what we would call an old-fashioned liberal. And was a Mower's course, the first course I took, was really the history of his life and really the history of genetics since the rediscovery of Mendel's laws.
He was a student of Morgan, right?
Yes.
I think, right.
And Morgan was sort of from Kentucky and highly conservative. And Morgan thought he was, being discriminated against. Probably true. The other of the famous trio of Maurer, Bridges, and Sturtevant. Bridges was very, very bright. I'm sure he was as bright as Maurer. And Sturtevant, who was at Caltech, was really, by the time I met him, just a boring old man and really not interesting.
And one should avoid boring people, right? We get back to this.
I go back to it, but so I had wanted to go to Caltech, but Caltech had turned me down because they had looked at my academic record, and it was largely courses about natural history and really very little chemistry. So I didn't look like a good person to, be interested in biology at the chemical level. But Indiana turned out to have the dean was very bright. It was at the end of the war, and American universities were still anti-Semitic, reluctant to put Jews on the faculty, even when they were, better than anyone else out of Europe. And Fernandez Payne hired 3 Jews instead of 1.
There's one person that I would like you to talk about. You met in Indiana, because we all know him, of course, in Italy, and he is Renato Dulbecco. He was actually your office mate, I guess.
Yes, and you didn't know what Renato thought. I mean, he was
he knew, probably.
Yeah, but he was, more reserved than Luria. He was certainly equally intelligent. Perhaps more intelligent than Luria. Luria was governed by emotions.
And this is bad? Is this bad?
Yes.
Okay.
Well, he believed that, the best scientists were, physicists, and people like Fermi. And then there were geneticists. And then he just thought chemists were boring. But solutions to all our problems were boring. Right, to Luria. So he never got interested in DNA because he did not want to think chemistry. So, I mean, that's what I'm saying. Emotions can get in the way of highly, and we can come back to it later. Why did Rosalind Franklin dislike helices? Totally and absolutely irrational. And yet it kept her from pursuing a helical structure. So and I almost acted irrationally because I met Erwin Chargaff. And he was an intensely unpleasant man.
We actually have a picture of Chargaff here, I think.
Yeah, there he is, looking grim.
Right. Looking grim, right.
Grim, a real grim man. And so I didn't want to believe that the base pairs were equal because his A and T were good. The G and C he was underestimating C. So you could say he didn't really prove 1-to-1. But Chargaff's unpleasantness actually did work to my favor, because in the summer of 1952, Linus Pauling was given his passport back, and so could come to the International Biochemical Congress. And at the Congress came Chargaff, and they were both speakers. And then they were scheduled and were on the same boat traveling back from France to the United States. Pauling decided that Chargaff was unpleasant and he wouldn't talk to him. So if he had talked to him, the only thing he could have talked to him about would have been A equals T and G is C. Right. So Pauling wrote a paper about DNA which never mentioned anything about the basis. So that was, you know,
disaster.
Yeah, but again, that shows you how, to the extent of where our likes and dislikes can sometimes have very serious irrational context, that we don't want to believe things by people we don't like.
Right.
And, you know, it's,
but we're jumping ahead a little bit.
we're
so
jumping ahead, but
yeah,
I'm just trying to come to that Luria, you know, if you said, well, afterwards, Luria did not have a distinguished career. I mean, in the sense his lab discovered nothing, whereas Delvecco really was very, very key in developing the field of tumor viruses. So, in that sense, you know, I think Luria was hurt by, he proved to be a very good department chairman at MIT.
An administrator, so to speak, yeah.
Yes, well, he was a nice man, and so people liked him. You know, Luria, you know, with time I realized, though, I didn't like Luria's friends. Because they were all left-wing. They were terrible people. I mean, extreme left-wing, people like Noam Chomsky. You know, I was, but you know, there's no fun being in a room with Chomsky. That's all I can try and tell you. You know, they sort of exude hatred of the United States, and you know, I think we're the greatest country in the world, still are. So you know, we have.
But speaking of people who like other people, this is somebody whom you met in Naples, right? And I would like you to tell us how. Who apparently didn't like you, right?
Yeah,
so Maurice Wilkins.
Yeah, and that worked against him.
So how right worked against him? But well, tell us the way you actually met him in Naples.
Well, there was a meeting on macromolecules, and I don't know whose idea it was. You know, only half the speakers were, about half the speakers were quite good. The other half were, you know, you had to invite this person and this person, and they were boring. But he'd invited the head of the biophysics lab in London, or whoever did the invitation, John Randall. And Randall didn't come, and they sent Wilkins. And Wilkins, just before coming, had obtained a much better diffraction pattern of DNA than had been obtained before the war by Asbury, and showed it for the first time in public. And interestingly, when he wrote up his article, he never put in the picture. Because I think he realized that putting a picture in would bring in competition. Because when I met him, I immediately wanted to leave Copenhagen and work on DNA. Because we had sort of the general belief that there were sort of sloppy structures. They didn't have a precise structure.
Not a crystal.
Yeah, and suddenly the fact that there was a crystalline X-ray to photograph said there was a defined structure. And you know, if you ask yourself how, and your real problem is how do you copy it? You got to know what you're copying. You got to know the three-dimensional structure. So, but Wilkins then, and I didn't know it when I wrote the double helix, so I sort of implied that Wilkins didn't want me. But then, because I went back to Copenhagen.
You denied it.
And wrote, I wrote Luria that I'd seen this X-ray photograph of DNA, but I couldn't go to London, and could he find me another place? And luckily he met the young English chemist John Kendrew at a meeting in the University of Michigan, and Kendrew was working alone and wanted someone to help him. And so, without knowing anything about me, but you know, figuring Luria wouldn't have a stupid student, they said they would take me. or he did. So then, so Laurie raised
incidentally, since you're talking about Kendrew, maybe you can tell us something about the Gran Paradiso, since we're going tomorrow, right?
Yes.
And the reason we're going tomorrow is because you went there with him, right?
I went there with John.
Two young people who would actually get the Nobel Prize the same day, many years later, right, for different reasons.
Yes.
So,
but the Gran Paradiso was sort of just exotic enough and different. And so John Kendrew wanted to go there, but he didn't want to go there alone. And so I was in the convent at the time, so I joined him there. In Konyan, we walked up to a hut, and then the next morning he suddenly tells me he's got to go back to Cambridge. And he didn't tell me why. It turned out later that his graduate student Hugh Huxley had just eloped with his wife.
What was the reason to go back then?
Well,
He was free then.
Because I think John wanted her to disappear.
So I see. He was trying to fight.
But anyway, so I never saw the Gran Paradiso. I just had walked out.
Okay.
So
we're going tomorrow, and we'll see if you bring luck to the people who will come with you, right?
But I remember I was drinking grappa.
Oh yeah, okay. We'll try to get you grappa.
Yes. But
let's go back to Cambridge now, right?
Yes.
And of course, the main person that you met in Cambridge is Francis Crick, and we have a little video clip that we can see, so that we have here Jim Watson in flesh, but we have also Francis Crick at least in pictures. So let's see this short video clip.
I want to take you back to the 50s, and I want to take you back to the first time you shook hands. And I'd like to see if you can recall what the energy was like. Did something tell you at that moment that this was going to be a Nobel Prize-winning collaborative or a great friendship?
No, of course not. No, I don't think I remember it. What I remember was going home, no deal saying to me, that's Max Pruitz, was round here with a young American, and you know what she said? He had no hair. I mean, he had a crew cut. So that's when I first sort of heard of Jim. But when we actually met, no, I think it took some time before when we got started talking and exchanging ideas, that really the sparks came, as it were.
I think we sort of established a sort of identity of interest. I was very interested in DNA, and most people didn't sort of share the enthusiasm. This perhaps was the most interesting problem of all, and Francis could see that very well it might be. And I arrived there just at a time when the alpha helix had been found, and Max Pruitz had got a good experimental demonstration that was right, and that Pauling had guessed the structure of the polypeptide chain by thinking in terms of its underlying chemistry. So we thought, well, could you do the same for DNA? I mean, it was natural. I think any two people who were struck by Pauling's success would say, could you do it also for DNA?
But I think it is because we both had although I was a beginner, we both had a broad interest in the problems. I mean, we focused on, the problems of genetics and what was the genetic material, what it does, and all the rest of it. And we'd done that quite independently. But that was rather different from most of our colleagues who were focused on protein structure and not looking at it from this wider point of view. You probably know the story of when Max Pruitz and John Kendrew found they had an extra room. They didn't know what to do with it. And they came, after a couple of days thinking, they came in and said, we're going to put you and Jim in that room so you can talk to each other and not disturb the rest of us.
It was pretty lively.
Yes. It went up and down. I mean, sometimes we didn't say anything, sometimes we talked.
Mm-hmm. What happens to the ego in situations like that, where you'll have an idea and you disagree, or you have an idea and you disagree, and your exchange gets to the point where they want to put you in your own room and close the door? How do you feel about each other in the process when you're having an argument, which might be the wrong word, but having that kind of discussion from different points of view?
Well, the point is absolutely essential to have that sort of discussion. That's the whole point of collaboration. When one person gets gets an idea, the other person, unless he's had it independently, he's going to show that idea isn't a good one. And it's only if you can interact in that way that you get collaboration. If you get a senior person and a junior person, and especially in countries like Japan, such a dialogue is very difficult indeed. And that was what was missing in London with Rosalind Franklin and Maurice Wilkins, and that's what we had in Cambridge. Wouldn't you agree with that?
Yes.
Certainly it's a collaboration that worked out, right? But when you started your book on the discovery of the double helix, the first phrase, which became famous, right, was, "I've never seen Francis Crick in a modest mood." So did you also get along as human beings, or?
Yes, extremely well. But, you know, most people have a lot to be modest about. Francis, you know, was really pretty unique, and we had no reason to be modest. I mean, the reason those of us, you know, who really liked to be near Francis was he was so often correct, and if you say anything sloppy, he would correct you. And so conversations with Francis were always sort of fun. And, you know, he would tell me I was being silly, and because sometimes I would say things that were so clearly false on purpose to see if he would believe that I believed them.
Just to see him react?
Yes, and Francis has, I think you could say, a limited sense of humor. You know, I think he didn't see the humor in his own behavior, you know. But he was the first person I could speak to about DNA. I mean, you met other people, but they didn't see the problem as information. And they didn't want to. For historical reasons, the problem didn't appeal to them. So, and then when I got to Cambridge, neither Perutz or Kendrew we had the slightest interest in the DNA problem. And, you know, they were focused, they both were sort of racing each other to find a three-dimensional structure of a protein. And I think Crick and I were different, and we didn't say protein structure wasn't, you know, boring or dull or anything like that. It would, you know, deserve a lot of Nobel Prizes. A recent one, you know, last one 2 days ago was for protein structure. And, but that in the, you know, rank of things, the molecule which had the information in that sense was more important than the one that provided the information too. So to us, you know, the secret of life would lie in DNA, and you would never understand what life was as long as you concentrated on proteins. You had to go to DNA, and that was just not accepted by people.
But this is a person, though, it's a picture of Pauling, who, of course, you know, certainly thought that DNA was important. What happened to him, though?
Well,
did he have a sense of humor, first of all?
Not much. And he was 50 years old, you know, in that picture.
No, I didn't know.
No, but that's roughly, he found the alpha helix, this great triumph, roughly when he was 50.
And then he wrote this, right?
And then he wrote the bad paper. But he'd been thinking about protein structure since the end of the war, so it had been a recurrent theme. He had really never focused on DNA, but had written a paper in 1939 with Delbruck. In fact, it was Pauling's idea. He said, he went to Delbruck and said, I want you to sign this paper because it attacks a famous physicist who says there's really an attractive force out of quantum mechanics between molecules. And he said, no, Pauling said it's always going to be complementary, not plus the negative, not like to like. And it was Pascal Jordan, who was sort of, apparently, those who knew him, very bright. Though, you know, he stayed with the Nazis, so his reputation naturally suffered. And Pauling wrote this paper, and then he gave a talk in Manchester in something like 1948. And he came out again, and he liked this idea that replication had to go by complementary. And it was, you know, a short published piece. This is the way genes are going to go. And yet by 5 years later, he wrote a paper on DNA which never mentioned complementarity.
Which is this one, I guess, right? A proposed structure for nuclear.
So it was.
Can I just note one thing? That apparently, if you wrote a paper on DNA in those years, you would get the Nobel Prize, even if it was wrong, because I see Pauling and Corey, they both got the Nobel Prize, right? But not for this reason.
Corey was a really very unimaginative technician. He was what we now call PhD technicians. And so he calculated coordinates for Pauling, and would have never disagreed with Professor Pauling on anything. In fact, Linus was not generally liked at Caltech by the chemists. He was admired, but
also for his political ideas, I guess, right?
No, I think Linus himself was not a nice man.
Okay, so more general. Even.
No, he did not want to. He wanted to do everything himself. He didn't really want to share credit. And so we had cases of rivalries at the time in Cambridge, where Pauling was just angry that people were, you know, slightly ahead of him. So instead of sort of congratulating Proust for a proof of Pauling's alpha helix, you know, he just wrote a slightly bitchy thing. And so I think he was that way at Caltech. He was nice to his children, but he really wasn't very nice to his colleagues, especially in the department. So you know, they just knew it was pointless to disagree with him. So he didn't get any feedback from his people. He had a series of reverential undergraduates. You know, he was a great teacher. And he had some, at the time, very bright people who did their PhDs with him. And they liked him. But he didn't like competitors elsewhere. And came to Cambridge in the summer of '52, and the alpha helix was a product of pure chemistry by ignoring the X-ray data. X-ray data, which the Cambridge people used, led you to the wrong structure. And it was because the alpha helices were coiled around each other. And Crick was worrying about that from the beginning. The moment I saw Crick first, he thinking in coiled coils. And Pauling showed up before the Biochemical Congress, and Francis told him his coiled coil idea. And as soon as Pauling got back to Caltech, he wrote a paper on coiled coils, sent it to Nature, and gave no credit at all to Crick for talking to him. Yeah, so, you know, I'd call that a crook. Okay. Right. And so the Cambridge people objected, and in fact, Pauling's paper in Nature came out several months after Crick. Crick was given the priority for it. But, you know, in that sense, you say Pauling was a great chemist and in many ways a very great man, but he was not a great human being.
Well, it seems that the story of the DNA discovery, the double helix discovery, is full of bad characters, right?
No, I mean, Pauling...
At least the way you say it, right?
No, the Pauling...
Chargaff was boring, then Pauling was a bad man.
No, I mean, Pauling... You know, Pauling was, you know, he'd give a lecture and he'd jump up on the platform, and he was a showman. He was, you know, loved in Hollywood, you know, and he was against the war, and he was, you know, for favor of better relations with the Russians, and had gone to a couple of peace congresses, which is why the State Department, you know, he was never a communist, not the slightest, but they just thought he was not in the interest. But of course, there was a later, Laurier didn't get a passport. And there was
for political reasons, I guess.
Yes, for political reasons.
Yeah.
I think because he had lied. He said he was not a member of the Communist Party, and I think there were people who they said we were in the party and he was with
him,
it.
Right.
So I think that was his problem. Salva never complained about it, and he never had any problems getting NIH money. He had never had any problems getting American recognition. But the State Department just, didn't want, someone lying in. At that time, we didn't realize how effective the Russian spy ring was. They really, Alan Nunn-May and Pontecorvo, they really had infiltrated everything.
Speaking of spies...
So I'm just trying to say, and so it was interesting that when, I didn't know it until recently, but Wilkins, mild-mannered Wilkins, the whole time I was interacting on this, telephone was being tapped by MI6. Yeah, he was one of 6 people thought to be the Russian spy in the uranium effort, because he had been possibly briefly a member of the Communist Party, but associated with anti-war efforts.
Movement.
Yeah, and in that sense, Wilkins was a typical Englishman, still dominated by the horrors of World War I. You just, World War I had so killed off the essence of England that they just didn't want another war. But Wilkins, we still don't know why. We got a record of the man named Peter Wright, who wrote a book called Spycatcher. And he once came to talk to Francis. So why did he come to talk to Francis? So, and I had thought possibly that John Kendrew was could have been a communist agent, because he really had an intense dislike of the United States taking over as #1 power from England. And really, thought people like John Foster Dulles and were that. And so he was #2 to Solly Zuckerman. And there's no evidence he ever was. But I could see a motive in Kendrew, because it was expressed in one of the British feelings. The spy, the real reason why he didn't do it was no liking for communism at all. It was just dislike of the American government being too powerful. So that was the driving thing. And certainly Kendrew had that. It was partly symbolized by, the arrival of Linus Pauling. America taking yes.
But
and but on the same sense, John, you had to like Pauling in a way, or admire him. So John, I don't think anything in John's career was ever hurt by this anti-Americanism. I don't think it in any way affected anything, except he just did not like
the United States.
He did not like John Foster Dulles, but neither did I.
Good.
So there were many people who just...
So maybe you were also a spy, and this introduces it to the next picture, because here is a character, right, Rosalind Franklin, that's still missing in the story, and somehow connected with spying too, right? Although in a different way, right?
No, I mean, Rosalind was, the story is... it's a sad story, because she had a really difficulty in, making friends, and certainly at King's.
So a character problem.
Yes, and I don't. Initially, there were people in King's who just, you know, thought she was what the English have this rare number of people called the English upper middle class. Okay, English upper middle class is by any other standards in any other country upper class. Her family were, you know, a couple hundred years of bankers, and
so she was rich by family.
yeah, but she wasn't landowning.
Uh-huh.
So she wasn't aristocracy and she wasn't gentry. So they were called upper middle class.
Upper middle, right.
So they weren't really, and that's where Rosalind came out of. So she really knew interesting people. Armin had the opportunity, went to a very good school, St. Paul's, and did her PhD in Cambridge, but never learned X-ray crystallography. She used X-rays, but she was not a person in this British school of solving structures, which is what England did, was, you know, considerable, and dominated the field until World War II, a little afterwards. And so if she had actually been a crystallographer, she would have realized the significance of something called space group, which had been worked out by Stokes, who worked with Wilkins, gave her the space group. And then a famous meeting, which I don't have in my book, but learned that she went up to Oxford to ask Dorothy Hodgkin, who was the sort of most respected English crystallographer, a woman, got the Nobel Prize for vitamin B12 afterwards, for help with the significance of the space group. And Rosalind immediately learned that, I mean, Dorothy immediately realized, Rosalind doesn't know any crystallography. She should go home and just learn the basic facts, and basically told her to do it. So Rosalind was never followed it up. And we didn't know what it was. Crick learned about it 2 weeks before the structure, before we found the base pairs, and said it means two chains running in opposite directions.
This is the picture, right? Yeah.
This would not have given it. This was the crystalline form. The crystalline form told you the symmetry. These molecules are sort of floating free. There's more water. You don't get orientation between them. The other, they were really nicely crystalline packed. So I saw, you know, the tragedy of it is that Rosalind didn't know how to get help. Whereas, you know, if you're sort of born, you know, with no mathematical ability, you realize maybe you should be friends with someone who knows mathematics. And, you know, I knew this from Crick. There were certain types of math Crick couldn't do. So he had a friend who could do these maths. And so
who was? Kreisel?
Kreisel.
Yeah.
A famous Kreisel. Really could...
He was actually in Torino, too, for a long time. It was difficult to get him out. But anyway.
Yes. No, Kreisel was... Crick had something... He liked Kreisel, and the only word you could describe it, kinky. I mean, Kreisel was just awful. But, you know, you know about him anyway.
Yeah,
so
right, because he was a logician.
He's a very famous person. But Francis met him, they were both in the Admiralty.
Yeah, right.
And so...
They were working for the war, as you said before.
And
right?
so
in the same room.
Yes, and Francis realized that Kreisel could do math that he couldn't do, and so that was the start of their friendship. Where Rosalind didn't somehow, you know, felt she'd have to do everything by herself. You know, that you wouldn't really deserve something unless you did it by yourself.
And she was not interested in models, for example, building models, right?
No, and again, I don't think she liked to think in 3 dimensions. And I've got into deep trouble for saying this is a characteristic of women. No. I mean,
I just read.
No. It has a history. The history is that
we get to the controversial things later on, but
I will tell you something that's not controversial. The IQ test. The IQ test, at least the ones that we get in the United States, are so. They choose the problems so that men and women each come up with the quote 100 average. Okay, but women can do some things better than men, and men can do some things better than women.
For example?
Women are much better verbally.
Verbally.
Yes, and the men's is we can read maps. Yeah. Okay. Just that's it.
All right.
Yeah. And it goes back to saying, we were hunter-gatherers. The men had to go out. They had to have a sense of themselves in space.
Right, in space.
Okay.
But
Just that.
bidimensional space, though.
Yeah.
Or spherical space.
Yeah. And so I think that Rosalind was just weak in space, but she was very good analytically, and you could say algebra, but she didn't
so she couldn't read maps in this sense, right? I mean, the pictures that she actually took.
No, this picture is, simple enough. Francis was going to write Fourier guides for the bird watcher and so on, So you look at that picture, it immediately tells you what helix is. Rosalind knew this. It's just, she physically, I don't think, liked thinking in this way when she could do something else.
So this is the question that probably everybody asked you. So you think that she would have gotten the Nobel Prize in the end if she had lived, of course? I mean, she died in 58, I
No,
guess,
I don't think so, because...
You don't think so?
You generally don't give prizes for people who make mistakes.
Well...
So yes.
Right.
So, and... Wilkins basically got the prize for two reasons, I think. And we felt good about it. We weren't worrying about Rosalind because she was dead. One was he took the ape photograph. So he really was the first one to say there was a real three-dimensional structure. And then second was, just personally in our case, he showed me that photograph that evening. And, from that photograph, it was just 1 month to the answer. And after we had the answer, we invited Wilkins up to Cambridge to see it. And beforehand, Francis and I had agreed that we would ask Wilkins to be a third author in our paper. And I don't know why I forgot and didn't put it in the double helix, but Wilkins puts it in his own autobiography.
What Map Pursuit, right?
That he greatly regretted that he didn't put his name on. Because, he really, that afternoon and evening, and when we were talking about this picture, he told me it repeats after 10 times, and I said, well, you got to go and build a model. And he said, no, I'm not going to build a model until Rosalind leaves the lab in 2.5 months. And I said, well, what is we're going to do with Pauling? Someone shows Pauling that he's wrong, and he gets to work and gets the right structure. We cannot afford to wait. And so it was easy to convince Sir Lawrence Bragg that we should be allowed to work on it because it wasn't just King's competing with Cambridge.
Right.
It was Caltech.
England against
it was England.
United States.
So England had to win, and therefore he wanted to unleash us. And so Wilkins came up. And then we asked him, do you mind if we build models? And Wilkins, every bit, he knew exactly what he had to say. He had to say yes, because he couldn't say no. But of course, he didn't want us to build.
And he said no?
No, he said yes.
Oh.
But, if he'd said no, we'd have done it anyways.
So, what's the reason to ask?
No, because, again, we did not Pauling to win.
But why did you ask him? I mean, if you had done it anyway.
Because he was a friend.
Oh, I see.
No. Both Crick and Wilkins were genuine middle-class people. They were taught up of good behavior. And I was sort of taught good behavior, but I, you know.
You didn't learn.
No, but
you were taught but didn't learn.
No, but I was in a financially deprived environment where manners, you know, were less occasion to be expressed.
Right.
So, you know,
but speaking of names,
but you see, at that time, when we said to Wilkins didn't know that Rosalind had actually looked at the bee thing, went back to London and discovered that Rosalind had lots of confirmatory data and had analyzed her picture, and that she was going to write a paper in any case, and therefore he had to show he had to write a paper, so he wrote a paper. And I think that he got the Nobel Prize was really Sir Lawrence Bragg arguing that this was the right thing to have. If we'd gone to Stockholm without Wilkins, it would have been bad, because we liked Wilkins, you know. And
so he was a friend, yeah, as you said.
He was a friend, you know, even though initially he could say he made this big mistake in not taking me into his lab.
I think we have a picture of the Nobel Prize. No, we don't. Anyway.
Yeah, there's a picture of the all four of us toward the end.
But speaking of this paper, actually, because you said Wilkins didn't like to have his name on it, and then he regretted. But many people would wonder, how come is Watson and Crick and not Crick and Watson? I mean, it's not alphabetical order, so there's a reason, or?
I think there is a real reason. We flipped a coin, but it was for the second paper. And, you know, I should have. No, the first was, I found the base pairs. You know, I did. Now, admittedly, Francis could have found them, but Francis was not working on the problem until we found the base pairs. He was still plugging away at his very unimportant PhD thesis on sort of...
On crystallography, right?
On formal crystallography of hemoglobin. You know, it's something that's very, you know, in retrospect, there might have been some reason to start it, but, you know, there was nothing. So Francis was... you know, and Bragg wanted Crick to get his PhD thesis because Bragg did not want to ever hear Crick's voice again, because Crick had a dominant voice, and Bragg didn't like the idea that Crick had insulted him. You know,
speaking of good matters, right?
Well, Francis probably correctly said, you know, I told you this idea a couple of weeks ago, and you didn't remember it. But so complicated as to really what did happen. But
we got to the main actually day of the story, right? Which is the 28th of February of 1962. What happened that day? First in the morning and then at lunch.
Well, you know, Francis wouldn't come in on Saturdays till more like 10.
So it was a Saturday.
And so by that time I'd been in the office for an hour and had finally put the base pairs together. And Francis came in and immediately saw the symmetry, which I didn't see, that you could put either base pair at any point in along the helix. So it was a perfect structure. And that, you know, the base pairs demanded the chains run in opposite directions, which is what the space group from King's had said. So you could have, you know, started from the space group and then got a two-chain structure. Or you could have got a two-chain structure. I got the two-chain structure, and Francis did give me credit for it by saying I really had gone through the density data and said the data really said two chains, not three. And
density meaning Chargaff's rules?
No, the density, you know, the amount of material, you know, how much water was in the structure? Was it dry or was it wet? And the B form is a much wetter structure where the molecules really can rotate around their axis, whereas in the A form, less water and you got the packing problem or the packing complication, which Rosalind had noticed a little asymmetry, and Francis from the start said the slight difference when you might think the reflections should be equal and they're not equal. He said they just pack slightly differently. And that's it. And Francis was right. Francis understood crystallography. Rosalind didn't. And so
we said that, right.
No, I come back to it because she's pointed out as, she was a crystallographer. No, she worked with X-rays, but she had never come near solving any molecule. And, normally, that would be a sort of PhD level thing. You learn the field after a couple of years and you know the tricks and you could, in that stage, get a job.
So that Saturday morning, everything fell into place, so to speak,
and then,
right?
we went to lunch. And
so
you could say, how did the famous sentence, that we found the structure of life? Well, I made it up.
You made it up?
Yeah, no, I didn't write it down any place. It just struck me as extremely plausible that Francis, we were in a very high mood. How could you go in there, see people you've not known, without saying, we've done something terribly important? We'd had to be.
So you could have said it too. Why did you say it was Francis?
Well, because he knew the people.
Oh,
he was a sort of
You wouldn't dare telling them.
Yeah, the Eagle was, most of the people in the Eagle were 30. And the reason they were all older, was because of the war. A lot of people would come back after the war, and so, whereas normally the age group would have been 25, there were a lot of people who were in their 30s. So the Eagle was filled with, at lunchtime, with sort of, there was a Persian chemist, and, there were a series of regulars that, and Francis had this horrendous laugh, and there was this Persian Ishaq. He became a very good economist, really good. He had a big laugh, so, he and Francis loved to laugh at each other. And so Francis was, had a lot of, these sort of casual friends. So, it was just perfect to do it. And then, the weekend, we hadn't, we knew we had to go and build a model and actually show that we could build the model. And that took a couple of days the next week. I have a friend on Long Island now whose father was in Cambridge at that time, and a friend of Francis. And he remembers me going to the Renaldini's house, his parents' house. His parents remember it because Louis was probably only 1 or 2 years old when this thing was happening, and my telling them that we had made a big discovery. So it was so clear that it was a perfect structure, and I worried, could anything this beautiful not be true? And, there was no way, except everything fitted in.
Proof by beauty, so to speak.
No fudging of, there was just no fudging. Everything just, it was beautiful structure. Whereas Pauling, his awful 3-chain structure, you had to fudge for that. It had a lot of nasty contacts, whereas the double helix just liked to exist. That's all. So it had a fluid. It was nice. Then I had to tell the people in Paris, Jacob and Monod particularly, and Lvov. So I went to Paris next week, and Francis spent that is after we knew we could build the B form, Francis took up the A form of how the bases had to be tilted. And when I got back, he said he knew what the transition between B and A. So, I always at that time, enormous reverence for Francis, just in the way he could do what had to be done. He was clearly so much more accomplished than I was, but I was, chiefly there, that it was still my problem. You know, in the sense that I took the paper.
Since Naples, right?
Yeah, it was my problem, and you know, I could be the loser. You know, I would have. Whereas Francis had something else. I had nothing. But it didn't worry me. I mean, I wasn't, you know, worrying about it, but that was the reality of it. Then we decided that because of the King's paper describing the structure, we weren't going to see anything about the replication things, and wrote this sentence, "It has not escaped our notice." Right. Intending to write it, which we did about a month later, called "The Genetical Implications of the Structure of DNA." And that's the one I think we flipped the coin on. And so I was not very generous. I should have let Crick have that.
And it went the opposite way, right? So
yes.
This is a Crick and Watson, right?
No.
Even the second one is Watson and Crick?
No, I won the coin toss.
Oh, again. So
no,
this is unfair. An unfair coin toss.
No, the first time there was no coin toss.
Yeah, I understand.
Okay, the second time there was a coin toss. And then there was a Cold Spring Harbor paper, which I delivered. And my name went first, but I was part of the Cold Spring Harbor War. Then there was a paper in the Proceedings of the Royal Society A, which described how the structure was built.
And finally,
and that had Crick's name first, but I actually wrote the paper.
Oh, I see.
And I wrote the paper because Francis was finishing his thesis.
So he was still working on that.
Yeah, he was still working on it. Well, look, when there's something that's boring and it's hard to work on. So he, and then Francis had arranged to go to the United States. And so the story was always, you know, a good story that should be recorded. And in June, after the second paper was read by the physicist George Gamow.
Oh, Gamow.
Yeah, Gamow read the second paper and then wrote a letter to Francis and
which appears in the title of one of your books, right?
Not
Together
the title.
With Jeans,
Jeans, Girls, and Gamow. But no, Gamow had a code, and it was wrong, and it was an overlapping code.
But so this was much later, right?
No, Gamow's code was in June of 1953.
Already?
Oh, yeah. No, George was, you see, he was a friend of Delbruck back in the early days, and he had written a rather nasty, not nasty, but he had written these very good books, Mr. Tompkins books. Yeah. And was coming out with one in genetics, which really...
On quantum mechanics, was it, right?
They were, yes, his popular were sort of popularizing quantum mechanics. And then he got interested in life because he was a friend of Delbruck before the double helix. So Joe had been thinking, you know, there must be a way to go the information gets into polypeptide chains. So, and Joe was very, very bright. And his code was disproven by Francis fairly soon. There was some way that Francis could disprove it. But, you know, Teller was trying to get his friends to make code.
But was it triplets already?
What?
Was it already triplets?
No, it had to be triplets because there were 20 amino acids, and 2 gets you only 16. But then you have to...
Redundant.
You have redundant, and how do you handle the redundant problem? And so Gamow, you know, until we began worrying about the mechanism of protein synthesis, when Francis had his adapter hypothesis, which was very important and correct. And then Sidney and I independently found messenger RNA. Then everything came together.
Sidney would be Brenner.
Sidney Brenner, yes. Yes. So the sort of DNA to RNA to protein with ribosomes as factories for protein synthesis. Francis's last really important idea in biology was realizing that RNA had to come first.
The central dogma, right?
No, the central dogma was DNA goes to RNA to protein, which
And
is
then-
me.
right.
Francis gets the credit for it, but
Oh.
it's me. I haven't.
So you didn't publish it?
Yeah, that
Because
was
it's
a
usually
mistake.
credited 1959 Crick, right? And
No. I was talking about DNA that goes to RNA before
Oh.
The double helix was found.
oh, even that.
Yeah. No, I mean, it was obvious. The
But since you're an atheist, you couldn't proclaim a dogma, right? So that's why.
No, I wouldn't have used the word dogma,
Right.
and Francis loved the word dogma. But his last really good idea was that RNA, if DNA makes RNA had to be there the first, because you could imagine sort of some RNA and then proteins and some simple
Direct.
form, and then DNA being chemically more stable would come later. And that is right was then in the early 80s, Tomczyk and Sidney Altman found evidence that RNA had true enzymatic capacity. And the final real proof of the importance of RNA was when the structures of ribosomes were solved. They found that the enzyme that made the peptide bond was a ribosome. The peptide bonds are made where there's no proteins, just RNA.
Yeah.
So,
yeah.
that part of it is absolutely clear.
But you never published anything about the dogma, right? Or the ideas of the dogma.
No, Francis published the dogma in a paper.
And didn't give you credit, though.
No. No, no, it was my fault.
Because you didn't publish it, right? So.
Yeah, I didn't, I wasn't, all I was worried about was trying to get the structure of RNA, which was,
Right.
So I had 3 years of failure. And that's sort of why the book Genes, Girls, and Gamma is not a happy book, but is...
Right.
Yeah.
Wandering around, right? Without knowing what to do.
But it had all Joe Gamma's letters and...
the type.
And, I was the only person at that time who really knew all the facts, and I wrote them up. And it's my hope that maybe, 100 years from now, people will say it's actually, well written, But the book never had a good opening sentence. And, I kept trying to find an opening sentence.
There's one thing that I would like people to see, because at this stage it's pretty clear. I mean, we all knew it even in advance, that the structure of the DNA is one of the landmarks of science in the 1900s, right? And this is a clip, a video clip, in which you and Crick compare yourself to other scientists like Einstein and Newton, and it's quite interesting to see, to hear what you say. So let's see if it works.
Yes, I agree. We wouldn't be in the same league, and I would also say of scientists, somebody like Einstein is quite a different level to either of us.
Why? Why is that true? Why? We're spending time, and it's delicious time in talking with you. And CBS was here yesterday interviewing you, and you're celebrated throughout the world for your contributions. Why is it that you're not in the same league with Einstein and Mozart?
Because we're not clever enough.
No, I think, we're very famous because DNA is such a wonderful molecule.
Yes,
and we talk
sure.
about it. But, when I think about how science must have been done 100 years ago or 200 years ago, I just think Newton was a lot more remarkable than we are, in the sense of, considering what he did. And these people, without lots of other people, to talk to, or science, and inventing a whole method. So I think they had a tougher job than we did.
Well, I think you wanted to ask, suppose something had happened and we'd been killed in a car accident. The structure of DNA was there. It was bound to come out.
Within 2 years.
Yes, 2 or 3 years or something like that. If you look at Einstein's theory of the photoelectric effect, it isn't at all clear that that would have been spotted within as short a time as that. And as for relativity, it was a totally new approach to things.
So, some people, are set something ahead 50 years or 100 years. And, I think We set it ahead 1 or 2 years.
This is an interesting hierarchy that you set up. Namely, the value of a scientific discovery is how much it precedes the eventual discovery by some other people. And you said, you know, if you hadn't done it, 2 or 3 years, the DNA would have been discovered anyway, right? Whereas for Einstein, for example, you know, general relativity could have been, you know, 50 years or 100 years. Is this the idea?
Is that actually true for Einstein? I thought there were people sort of laughing at that sort of...
The special relativity, but general relativity probably...
needed Einstein.
Right.
But is general relativity as important as special?
Well, don't ask me, of course, right?
No. I'm trying to...
It's probably more important, at least for GPS, right?
I mean, you know, I've sort of heard, you know, that you know, Einstein spent the last of his life trying to really prove it, and he never could.
No, he actually was trying to unify all possible theories,
yes.
so he didn't succeed in that. But the general relativity certainly, you know, is one of the landmarks again, right?
Yeah, okay, we never thought of...
of yourself as the same standard, right?
No, I think, you know, so in biology we can sort of see where we stand, and you know, there's probably no one between us and Darwin. You know, in the sense that the DNA really did change things, and of course,
even the perspective of, on evolution.
Yes.
And so you think it provided a proof, actually, of the theory of evolution?
No, it was proven without us.
No, but I mean the mechanism.
Yes, sure.
Yeah.
But in terms of, you know, giving a chemical basis to biology.
The biological mechanism.
So, you know, I mean, if you try to think, the geneticist that was at Indiana, Muller, he was perhaps the greatest of them, you know, between Darwin.
Between you and Darwin, right?
Yeah, I would say because he really had a very good mind. He was really saw the...
More than Morgan, for example.
Yeah, Morgan was Morgan was not an intellect. Muller was a real intellect, and you know, and Morgan was a Republican.
So?
No, I'm just trying to say he was a sort of conservative man. He came out of a Kentucky colonel. He was just not, you know.
Oh no, I perfectly understand. In Italian, we would say he was a Berlusconian, right, or something like that.
Whereas Muller was really saw the implications of genetics and was just, I would say, much more interesting.
I'm afraid, I mean, they sent me a note that we only have a few minutes left. So, of course, you know, we concentrated our discussion on the history of the discovery of the double helix structure. And then we should jump 50 or 60 years, right? And I know that you're in Cold Spring, of course, when you went there in 68, you started developing a research on cancer, right? Yesterday you've been to Candiolo, right?
Yes.
Could you tell us what's new in this field and what are the hopes to actually cure what people thought would be incurable cancer?
Yeah, that... We've been searching using genetics and biochemistry to find what we call the drivers of the cancer cell. What makes the cancer cell grow out of control? And then it's turning out that for many cancer cells, they have a number of drivers. Some are more important, but so we've been trying to produce drugs against the drivers. And part of the problem is that a cell can mutate or in various ways change its driver. So you inhibit it, you stop one driver, and then boom, another driver takes over. And so there's some of us who think that you really might be better to ask the question, what is the greatest vulnerability of the cell? Which molecules does it need the most? And go for vulnerability, and you do this by another DNA-based method, but it uses inhibitory RNAi, and so we're searching for vulnerabilities.
Mm-hmm.
And there's a reason to believe that cancer cells, because they're growing out of control, have inherent weaknesses in their biochemistry, and we're trying to find them. So I'd say that is...
But is there already some practical results, like things, for example, that could go on the market in a near soon?
We finally have, and it was got through the vulnerability thing. Within a year, there'll be a drug against the very main driver, MYC. So we'll see, and that may, by taking out MYC, it may actually cure cancer by differentiating and not killing it, the cancer cell, but in the way somehow the cancer arose, it just differentiates to a macrophage. So we're going to see.
Okay.
So, but what's clear is that I think certainly the American effort is totally out of balance, just sequence, and no one is producing the drugs.
That's the problem, right.
So we can give genetic diagnosis and say your cancer has this, but it's not going to cure you. But they're filling American TV, but the public is being brainwashed that the cancer problem will disappear because we're sequencing DNA. And it won't. It'll stay just where it is now. I'm trying to
read the sequencing.
We're just trying to find vulnerabilities, and there. In my talk yesterday, I tried to give a direction. It comes up with a slightly, for many people, surprising conclusion that the real enemy are antioxidants, and the good things are oxidants. So all those who just think that because you're supplementing your diet with antioxidants, you won't get cancer. In fact, you may just be doing the opposite. So cancer cells fill up with antioxidants so they can't be killed. This is maybe right or wrong. It's an easily testable idea. We'll know in a couple of years, maybe sooner. But I'm convinced it's right because it's just...
too beautiful to be wrong.
No, there's a lot of evidence. The cancer world, because it's complicated, has had the luxury of never having to decide whether something is right or wrong. They just, some people say it's right, but they never really come to the grips and say, this is right. They just think everyone is different, and so they're slightly afraid to generalize because we're all so different, and the cancers are all so different. They're providing a lot of excuses for failure. And so compared to, the days of Francis Crick and the early days of genetic code, the cancer world is all they're doing is raising money and trying to get their papers published and not thinking.
Okay.
Yeah, I mean that's I know it's unfair, and I want also, but the caveat is the pure cancer research we've done is of very high quality, and it's beautiful pieces of science. It's not now totally focused on curing cancer.
Okay.
It's sort of enamored so it doesn't have the feeling, whereas I'm at 85, I know people, they've been treated with cancer, it's in remission, it's going to come back and there's no drug that's going to help them. Okay, I don't like that. Right? So
this has to change.
So yeah, I think, but if there's one thing you discover, it's almost impossible to get someone to give up their own ideas because you think yours are better. They will go ahead with what they're doing. So if you want to prove your idea, you got to do it yourself. Don't expect other people to. And
this could be one of your famous sentences, right? Like the ones you put in in your book, right?
Well, but I'm actually very satisfied with what cancer research, and I think it's yielded. Some very, very high quality science. I learned some, things I didn't know yesterday. Beautiful science. So, it makes me think. And so I don't think we have to go on for 50 more years. And that's my thought, but we should at least say we're going to do it in 10. Because if you said you're going to do it in 10, then...
You could do it in 20.
Yeah, then you would immediately realize there's no way we're going to do it in 10 unless we hire some more chemists. Right. That's all. So, we're proceeding at a rate where the sort of number of people who die of incurable cancer is going to be about the same 20 years from now as it is today, unless we just...
Shift. Right.
That's all. And, but, the hope will be trying to find, people aged 30, 35.
That have big dreams, as you say once, right?
Big dreams, and they're not committed to something else.
Right.
And,
so somebody like you,
there's something, you go into a group of people, someone here, or certainly that group I was with at noon, and just think, what a large number of really bright people. And then you see them at age 40, and they're so boring.
Right, and you should avoid them, right?
No, what I mean is that,
I understand.
that they're not at 40 working on something important.
Right.
That's all.
Right.
That somehow the dream of, of accomplishing something big.
So they should do it early when they're 20 and think big, so to speak, right?
Yeah, at least try and do it big. And so you've got to have a society where, Crick was older then. He was 35, but that was because of the war. Right. Ordinarily, you would be dealing with, I'd say the group between 25 and 35.
Se non ci sono domande, I'm afraid you'll have to...
Sì,